Important topic, which is nerve muscle physiology. In the nerve muscle physiology, we'll be talking about resting membrane potential. Resting membrane potential is always negative, and the reason is because of the continuous efflux of the potassium. Because this potassium is leaking out of the cell, if this is a cell, potassium keeps on leaking during repolarization. So potassium goes outside the cell. Because of that, there is a negative RMP. Now the equilibrium potential of the potassium, it is minus 90 millivolt, and because of this reason the potassium tries to be in equilibrium, and that is the reason we have a RMP which is a negative. For a neuron on a nerve, the RMP is minus 70 millivolt. For a skeletal muscle, it is minus 90 millivolt. For cardiac muscle is also minus 90 millivolt. For SA node, it is minus 60 to minus 40 millivolt. For GI cells like Kajal cells or pacemaker, again it is minus 60 to 40. And pre-Bodzinger complex, it is minus 60 to minus 40. So RMP of the neuron, which is most frequently asked, it is minus 70 millivolt. Now equilibrium potential of certain ions. Sodium has got a equilibrium potential of plus 60 millivolt. And that is why when we talk about action potential, initially there is sodium that is coming inside the cell. And that is the reason inside the cell membrane becomes positive. And therefore there is a rise or peak in the action potential which is seen because of the positiveness. So when the sodium comes in, because the sodium has got an equilibrium potential of plus 60 millivolt, that is why when lot of sodium comes in but not reaches the equilibrium, it is slightly less than the equilibrium. So when the sodium comes in, the cell membrane is positively charged. Sodium has got an equilibrium potential of plus 60. Potassium is minus 90 millivolt. Because the sodium exits the cell, that is why the RMP of a cell is mostly negative. That is because of the exit of the potassium. Chloride is minus 70 millivolt, which is exactly the RMP of a neuron. Calcium plus 130 millivolt. Equilibrium potential or isoelectric potential, it is calculated by Nernst equation. So isoelectric potential is given by the Nernst equation. Now membrane excitation and impulse transmission. So membrane excitation occur in two phases or two forms. It can be electrotonic conduction or it can be action potential propagation. A membrane can be excited via two ways. Either there is a conduction of the ion that is responsible for the membrane to excite, or there can be action potential, and because of this membrane can excite. So let's first talk about the electrotonic conduction. Now what happens? There is a direct and positive charge spread from the point of entry. So there is sodium that is coming inside the cell. Now what happens? There is also the charges that leak because of the membrane capacitance. The sodium is coming in, but because of the membrane capacitance which is leaking out, or membrane resistance is low, the strength of the signal is weak, so the charges, they also leak out in between this potassium sodium pump. We have these charges that are leaking, and such a transmission in which charges are leaking and initially there is more positive, then after some time there is less positive left. Say suppose initially if there was plus we can say plus 30 or plus 35, then the positive ion starts leaking. Then what happens? After some times it goes to 0 or say minus 50. When it goes to such a low level, then we have these charges that are the positive charges, they are leaking out because of the membrane capacitance, and we have a positive charge which is going down, and such a transmission is known as decremental because it is decreasing. The positive charge is decreasing in length as there is a conduction. So because of this electrotonic conduction, the charges are leaking out and the condition or transmission is decremental. The carry impulse only up to 10 millimeter. After 10 millimeter of length in the neuron, there, because of this decrement, there is no charge left for the next impulse to be generated. So from dendrite, these are the dendrites, to axon hillock, this is the axon hillock, the charges travel by electrotonic conduction. So from here the charges are traveling by electrotonic conduction, and from the axon hillock we have saltatory conduction from nodes of Runway to nodes of Runway. So this is actually, saltatory conduction is just a misnomer. Charges are traveling in a straight line only. But because here are sodium pump present more concentrated at the nodes of Renvir, we have very more concentrated sodium pump. So lot of sodium comes in, and that is why there is a rise in the action potential, and that is why we get more positive here. And also because of this myelin sheath, there is no leakage of sodium, so this is also preserved. So that is why we say saltatory conduction. It seems like at the nodes of Runway the action potential is rising up, and that is why it is a misnomer of saltatory conduction. So initially at the axon hillock there is initiation of the action potential, and because of the enough number of sodium potassium channel, sodium channel, and because of lower threshold there is a rise of action potential at the axon hillock area. Then we have node to node conduction in the electrotonic conduction, and in the saltatory conduction now there are nodes of Runway present here, and we have very high number of sodium channels here at the nodes of Runway, and because of that more sodium comes in at the nodes of Runway. So and also there is no sodium or positive ion leaking out. It is not leaking because of the myelin sheath, because of the covering, and for this reason the leakage is preserved. So there is a long distance conduction from one node of Runway to another node of Runway because the ions are not leaking out. This is known as saltatory conduction. So electrotonic conduction is decremental because of the leakage of ion, while on the other hand saltatory conduction is not a decremental, it is not leaking out because the overlying myelin sheath is preventing it. But in case of electrotonic conduction, as the overlying myelin sheath decreases the capacitance and increases the resistance. So basically resistance is inversely proportional to capacitance. So this kind of conduction is seen in the retinal cells, in the rods, then the bipolar cells, and the ganglia. So in case of myelinated nerve, the sodium channels are present at the nodes of Runway, and they are present in very high concentration. So no sodium channel is beneath this myelin sheath. Sodium will travel a lot of distance since sodium is not leaking out. While on the other hand, in case of a unmyelinated nerve, there is no myelin sheath protecting it. So sodium channels are located at a regular interval. So this is a local circuitive flow in which the sodium is coming from the sodium channel, and we have this ions leaking out in between these sodium channels in the unmyelinated nerves. And that is why now you can remember also, if you put lot of brakes while driving, your driving will be slow. So here in the myelinated nerve, these brakes are coming because of the sodium entry at the nodes of Runway. Nodes of Runway are located at a far distance. So here the sodium is traveling very fast. Then there is slowdown because of the sodium influx into the membrane, and then fast, then slow down. So very less brakes it is putting. But here in case of unmyelinated nerve, very frequently if a car puts brake, then the speed slows down. That is why unmyelinated nerve has slow conduction rate. while on the other hand a myelinated nerve has a high conduction rate. So saltatory conduction, it is faster, while on the other hand the unmyelinated nerve, there is a slow conduction rate. Also this saltatory conduction or myelinated nerve consume less energy which is required for recharging, because recharging occur at node of Ranvier by the sodium potassium pump, and they are located at a far distance. So saltatory conduction in the myelinated axon results from the fact that voltage gated sodium channel, they are more concentrated at the nodes of Ranvier. Now coming to the second, this was about the electrotonic conduction, which is also known as graded conduction. Coming to the action potential, which is a very important topic. Action potential is also known as the self-regenerative process, because once this action potential is started, now if you remember in the Diwali, you can see this red color firecracker. You just have to light one firecracker from the whole chain. Then the chain automatically catches the fire. So if you remember this example, you just need to light one and all the cracker will start bursting. So all the chain will be bursted if you have not unwinded it. So likewise is the action potential. You just have to generate one action potential and it will generate another and it will generate another. That is why it is self-regenerative, also self-propagated process. So there is no decremental conduction in case of action potential. It obeys all or none phenomena or all or none law. If the action potential, if it is above the threshold level, then you cannot stop it. Either it is going to be plus 35, plus 25, any of it. But once you have crossed above the threshold, there is going to be a peak or rise, or there is going to be an action potential. If it is below threshold, then there is going to be no action potential. So once it is above the threshold, either it is going to be all, or if it is below the threshold, it is going to be no action potential. So it obeys the all or none law. It is not graded. So there are phases of action potential which is recorded by the cathode ray oscilloscope. So this is the graph. RMP of a neuron, it is minus 70 millivolt. So we start from minus 70 millivolt. Now what happens, there is sodium that is coming inside the, so sodium is coming inside the cell. So since earlier there was a negative RMP minus 70, because sodium is coming, the charge of the cell membranes is becoming positive. Now what happens, till the sodium comes, the membrane is going to be positive, positive, positive and more positive till it reaches the peak. Then what happens, potassium starts going out. Since potassium is also positive charge, so the positive charge start leaking, there is exit of the potassium, and that is the reason again the membrane comes to the negative side. All the sodium is coming in, so we have sodium potassium pump which takes the 3 sodium out and takes the 2 potassium in, and that is why we have this whole of the cycle in the process that balances the ions. Otherwise there will be more and more sodium inside the cell and cell is going to swell up and it will burst. So this is how a potassium ATPase pump is going to regulate the cell volume, and it will not let the cell burst, and that is what we study in the thyroid hormone also. So basically, now coming to the point again, the entry of sodium will initiate the action potential and the exit of potassium, efflux of the potassium will generate the repolarization. So depolarization is because of the sodium entry into the cells. Repolarization is because of the potassium exit from the cell. Now what happens? It is not a continuous process. What happens? At somewhere between, say suppose this is a point A, here threshold point B, then we have a point C here, and this is at top is the point D. So somewhere between point B and point C, the sodium entry stops. Now it is like if you have gone to watch a movie and there are only limited tickets. All the people, all the crowd is standing outside the movie theater. And a person goes and opened a gate. Another person goes and open another gate. Third person goes and open third gate. Now what happens? After 10 people, all the gates are open and all of a sudden, whole of the crowd gets in. Now from CCTV camera or surveillance, someone is watching. They close all the gate at a time. So the gate stops at somewhere between point B and point C. So, and after that, since so much of sodium has come in, there is a rise or spike in the action potential. So again, they can ask questions like this. Now what happens at somewhere point A and point B, the potassium exit will start. It starts at point between point A and point B, but the exit, potassium exit, is very small. So since there is a difference in the action potential, since there is a difference in the concentration gradient of the sodium, outside I guess it is 141 or 14. So all of a sudden a lot of sodium comes in, and that is why there is a spike or rise. So somewhere we have here, say suppose it is a 0. This is the upstroke up to 0 from threshold, and from 0 to plus 30 it rises up to plus 30, plus 35 actually, it rises up to plus 35. Then this is known as overshoot. This particular spike, which consists of depolarization as well as repolarization, it is combination of both. Then what happens after point D? There is a more potassium going outside the cell, and that is why there is a sudden spike or downfall in the repolarization. After a certain point, we have a term which is known as after depolarization. Now what happens? Since the gate are closed, but still a lot of potassium exits, so there is fall which is equivalent to the equilibrium potential of potassium. Potassium equilibrium potential is minus 90. So this graph falls up to minus 90. Potassium tries to be in equilibrium, that is why it falls up to minus 90. Then the RMP comes again back to minus 70 millivolt, and this fall up to minus 90 is known as hyperpolarization. So this was about the action potential. We have other ways of depolarization also. We know depolarization is because of the sodium influx. Apart from sodium, there can be calcium entry, which is seen in the heart. There can be potassium entry also. There is only one cell in the body which takes potassium. All the cells are going to exit the potassium, hair cell, which is going to take the potassium in. So there is accumulation of potassium inside the membrane, which is also seen in pancreatic beta cell. So spike is a rapid rise of action potential, which is depolarization, which is because of the sodium entry, and a rapid fall, which is because of the depolarization and efflux of the potassium out of the cell. Then we have after depolarization, a term which is after depolarization. So this particular point is after depolarization, where there is a downfall of the repolarization. Hyperpolarization is slow down of this repolarization and it becomes more negative. So depolarization, it is RMP to threshold. This is depolarization. From RMP to threshold, there is a positive feedback cycle of the sodium channel. So what happens? There is a stimulus. Then what happens? There are some sodium gates that open. Sodium start coming in. Then all of a sudden there is all the gates are open. A lot of sodium comes in. Then the membrane potential drops down. Then again this cycle continues until it reaches the threshold. Threshold is the critical point. So this is the threshold. It is the critical number of sodium channel when opens. Massive surge of the sodium that follows by the closure of the sodium channel. So one single push at the threshold and closure of the sodium channel will reach the action potential up to 35 millivolt. All of a sudden, a sudden entry of the sodium will cause the rise of this action potential up to 35 millivolt, and all of a sudden there is going to be closure of all the sodium gates. Then that will take it above the threshold. If the threshold is reached, we call it action potential. If the threshold is reached, then we say that there is a spike of the action potential. So this is about the action potential. Now we have some more terms like absolute refractive period. So this is the absolute refractive period in which there is no further action potentials that stimulate the nerve during this period. So from the start of threshold till 1/3 of the repolarization. From the start of this threshold up to 1/3 of this repolarization is the absolute refractive period. Even if you give more stimulus, there is nothing going to happen because the action potential will be same. So this is before the threshold. If you give more excitation, there will be generation of the action potential. Then we have relative refractive period. If you give, after this period is the relative refractive period. After this period, if you give the stimulus, then there will be next excitation potential. There will be generation of the next stimulus. So it will generate the next action potential. It starts from the 1/3 of the repolarization till the complete of the action potential. This is relative refractive period. So absolute refractive period is even if you give stimulus, nothing is going to happen because already the sodium, so much of sodium has come in and the gates are closed now. Even if you give stimulus, all the gates are closed, the cell membrane is fully loaded with the sodium. Action potential is going to rise up to 335 plus 35 millivolt. So depolarization is because of the sodium entry inside the cell. Repolarization is because of the potassium exit outside of the cell, and this is the peak action potential. Now compound action potential is seen in mixed nerve. Amplitude of the action potential can be increased maximally by increased number of open sodium channel. Action potential is produced because of the influx of the sodium. The permeability of the sodium ion increases during the depolarization of a nerve. Magnesium is not associated with the transmission of the nerve. Concentration of potassium determines the resting membrane potential on the nerve. Now, there is a previous question from chronaxie. For that, we have to understand what is a Rio base. So, minimum strength of a stimulus that can excite the nerve. It is Rio base. So, this is the minimum amount of stimulus that is required to reach up to the threshold so that action potential can be generated. And this chronaxie is the time taken by the tissue to excite when the stimulus strength is doubled the Rio base. So it measures the excitability of a cell. Increasing order of chronaxie is firstly seen in the myelinated nerve. There is a least chronaxie, it is most excitable. So more the excitability of a cell, lesser is the chronaxie. So it is the opposite. So myelinated nerve have got the least chronaxie, it is more excitable. Unmyelinated nerve has got, then second one we have unmyelinated nerve, which has got second most excitable and second most chronaxie. So minimum chronaxie is seen in the myelinated nerves. If not in the option, then unmyelinated nerve. If not in the option, then skeletal muscle. Then we have cardiac muscle. They can even ask you in a sequence based question or smooth muscle. So this is basically we are talking about membrane excitation. The most excitable membrane is the myelinated nerve. We have already talked about because there is no loss of sodium because of the myelin sheath which is present. So it has got a least chronaxie and it is the most excitable. So basically chronaxie is the time taken by the tissue to excite when the stimulus strength is double the Rio base. Rio base is the minimum strength of the stimulus that excites the tissue. And if we double it, the time that is taken to excite. So myelinated nerve is excited faster. That is why the time taken is very less, because it is excited very faster. So if the excitability is more, the time taken is very less. So for chronaxie, myelinated nerve has got the least because it is excited very fast. So the time is very less. So this is the order.